Yes, so ladies and gentlemen, I know everybody was very excited for this upcoming panel discussion. The topic is very relevant, and we have our panelists who I will be adding to the floor very shortly. The panel will be led by none other than Mr. Hugo Gagnon, CEO and VP of PhenoSwitch Bioscience. Our panelists, which you'll introduce shortly, will include Anka Hardy, Mukund Chorghade, Vivek Vishnudas, Dr. Hiroomi Tada, Joe Varshney, and Dr. Woody Sherman. So I will be adding all the panelists now simultaneously, and I'm sure everybody here will be enjoying this panel discussion. A lot of work has been done by our panelists. They've contributed the past 2 weeks with their ideas to deliver everybody a thoughtful panel, most importantly, and thought-provoking as well. So without any further ado, I'd like to give it up to Mr. Hugo Gagnon.
Thank you very much, Sam. So, I think I will start. I'm very pleased to be the panel moderator for this panel. We have, I would say, a very nice table of scientists passionate about what they do, and there's a lot of expertise around the table. So I hope everyone will have the chance to gain new knowledge about that. I prepared a few slides to support the presentation. So the first step of this panel will just for everyone to present themselves rapidly so everyone knows who does what. So I will start by myself. If you weren't there at the presentation earlier today, I'm the CEO and Vice President of PhenoSwitch Bioscience. In terms of new technology and early drug development, we apply omics expertise with data analysis, deep data analysis to help research and drug discovery. So who's next?
I could go next. I'm Mukund Chorghade. I'm currently the President and Chief Scientific Officer of a company called THINQ Pharma. THINQ has an interesting origin. I mean, its abbreviation is expanded into T for technology, health, innovation, novelty, and then Q for quality. I am a very dyed-in-the-wool synthetic organic chemist who has contributed to drug discovery and development by making of molecules, scaling them up to manufacturing scale. I have been a serial entrepreneur, and the current company I run focuses basically on the chemosynthetic liver and how it can be used to expedite drug development, in particular by detecting a lot of hidden metabolites. I also have faculty positions at many universities where I'm an adjunct. And when people always ask me, how come you have a foot in both doors, as it were, my answer has always been invariant, that if I'm in an academic group and I talk some incorrect things or even nonsense, I have a ready-made escape route by saying, well, I don't know, I'm from industry. And the same thing works in reverse as well.
All right.
Brilliant.
So who's the next one?
I could go next.
All right.
I'm Joe Varshney, founder and CEO of VeriSIM. A little bit of my background: I'm a veterinarian with PhD in comparative oncology and computer science genomics. I've been very passionate about translational sciences since, I can remember since I was a veterinarian. My passion really became the invention of VeriSIM Life, what we do here. And if you were in Victor's presentation, which was right before, basically we are an AI-enabled computational platform to simulate the physiological phenomena between human and animal systems to be able to make the early go, no-go decisions in early drug development pipeline. And we can discuss this more details, and also serve as strategic advisory board for several biotech and healthcare AI companies. Nice to meet you all, and I hope everyone is doing safe. Thanks.
Hi, maybe I can go next. I am Anka Ehrhardt. I'm a biophysicist, and nominally my PhD is actually in human physiology. I always say I do know how the body works, not how the mind works. And I do have very extensive industry experience. I came into industry really from the biophysics angle, running and building large technology labs with any kind of technology you can think of in biopharma industry and research. And with those labs, I started out in industry really supporting all phases from very early discovery. A large amount of my time I spent in target validation and screening. Then I really brought that over into the translational sciences, and now I am focusing a lot on clinical research using all my preclinical knowledge to really drive and connect between those different phases in the drug discovery and development. My current focus is on, I work for CHDI. We are a non-profit working on Huntington's disease, which is a deadly genetic neurodegenerative disease. And I am responsible for the clinical biomarker strategies, for clinical biomarkers, clinical samples, and anything that is related to research with them.
All right. There's Woody and Hiroomi.
I can go next.
Yeah.
Yep. Hi, everybody. Woody Sherman. I'm Chief Scientific Officer at Silicon Therapeutics. We're a Boston-based biotech company focused on primarily oncology targets. We built a physics-based platform to drive our drug discovery efforts, primarily based on quantum mechanics and molecular dynamics simulations and free energy calculations. My background has always been in physical chemistry and molecular simulations, and happy to be here. Happy to talk with you all.
Great.
Hey, thanks, Hugo. I'm Hiroomi Tada. I'm a Chief Medical Officer at Notable Labs. I'm a surgical oncologist by training, as well as having a molecular biology PhD. Notable is a biotech startup in which we've developed an automated technology platform that enables precision drug development in heme malignancies. We're using fresh patient samples to test for hundreds of different drugs and drug combinations, and really looking for personalized therapeutic options, and also using it as a drug development platform in partnership with pharmaceutical companies.
All right. I think everyone that is there already present themselves. So let's take a look at today's agenda. We will have a few poll questions to the audience that we will ideally reveal at the end for further discussion. Then we'll have a discussion, a roundtable with the panelists. After that, we return on the poll questions and results and discuss it. And I would like all the panelists at the end to make a closing remark based on what we discussed and their expertise in that field. Let's start with the poll questions. Sam, you will make them appear, I guess? Okay. So the first poll question, when it comes to integrating new innovative technologies, how do you prefer to integrate them? So the first response would be fully outsource them, collaboration with CROs or other academic groups or whatever, or fully develop internally. That are the three choice and answers. Please, I would like so people as much as possible can participate, so we don't have to have no answers at the end.
I could possibly answer that. So I'm a big believer in collaboration with a CRO, so that you have a hybrid mix of some internal resource as well as some external ones. You cannot just toss it over the fence at some CRO. Sooner or later, it will be very helpful if every member of the team in a company also understands these technologies, and by joint discussion makes things a lot better. Now, I fully understand that in the early discovery level, it is not always traditional to outsource everything anyway. The outsourcing mainly comes at the later stage development, where a new biotech frequently lacks the resources to do things like manufacturing and formulations and all. But I'm a big believer in the hybrid model, where you should do a little bit of both.
Thanks for your input. It's indeed my whole opinion also on this way. Can you pop out the second poll question? All right.
Does anybody else have anything to add to that? Maybe criticism of my answer?
I mean, I agree with Mukund here. I think for us, because we do this on a regular basis and adoption to AI and machine learning, perhaps every panelist here knows, and they have been there, done that, and is doing it. And one of the key things we have learned is we want to educate the scientists because they should be able to understand, because at the end of the day, they are the experts, we are not. And they're solving a biological problem. So, I think less of a criticism, it's more about that marriage of hybrid building, of how models can really integrate biology rather than having an AI standalone model that spits out biology. And then there's this silo where we don't have the expert who can interpret what AI did versus what the biologists are asking. And it has been quite successful for us. So I think we want to follow that model as long as we can and scale the company that way.
All right. Second question, did it appear? Yeah, it's there. So what do you think will affect the most early drug discovery in the coming years? A couple of choice of answer: new instrumentation, informatics that will include computer, AI, new methods, actually new research that will bring new things together, new ways of doing research. So let's think a little bit differently about how we do the research. New molecular biology tools. If we think of CRISPR, it's one that has been appeared in the last years. And real world data, so how we can use the real world data and apply it to research. So those are the choice of answer.
Coming in.
Again, may I just throw in one thought for you? I'm a big fan of the new methods and even CRISPR and all that. That is quite exquisite. I think you are going to see a phasing out of some of the older methods like combinatorial chemistry, which have been responsible for producing molecules with neither chirality nor diversity. But there is one real world data I really want to emphasize. I mean, it's thought about in a typical way. I feel over the next 2-3 years, the real world data derived from complementary and alternate medicine, traditional medicines from India, China, and all that will come pretty big into the fore. So there is a new field almost called as observational therapeutics, where these physicians have recorded patient data where they have administered a lot of herbs and other things to real patients, but they lack the power of chemistry or all these modern techniques that we talk about. But those observations are fairly valid, and I think this will be all nicely back integrated into our discovery process.
Yeah, that's a good point. Well, if I may comment on that, one thing we've seen, at least here in Quebec, is a big problem in informatization of the medical patient information. A lot of things have been scanned and manually entered, and it's just not usable. So that's something that actually I think pharmas and everyone would have a big gain just to help the healthcare system to get that up to date and be able to use that. Yeah.
Also, I think the Western pharma has always focused on one target, one drug, and one disease. Whereas I think in these forms of medicine with different, you can get multi-targeted drugs.
Yeah.
And I think some of this can be very useful in targeting antibiotic resistance and many other things.
All right. Third question. So where do you think innovative technology have the most impact during the course of your drug development? So lead identification, lead qualification, biomarker discovery, biomarker identification, speed of discovery overall, mechanisms of action understanding, or during clinical trials?
Maybe I can say something about that. For my opinion, I think a lot of impact will come in the discovery of new targets and new biomarkers, because right now we are seeing the technologies becoming ready for all the omics and for handling the data from the omics, mining them for discovering new insights. So my vote would be really on the discovery both of mechanism of action insights as well as biomarker discovery.
Yeah, this is the hope for personalized medicine at some point, when you are able to early identify biomarkers or new mechanisms of action, or segregate something in the model and be able to apply it on the clinical thereafter.
I have to agree with you, Anka. I think this MOA thing is going to be very bigger, get even bigger, and so on.
All right, and fourth and last question. In your organization, how long does it take to implement a new technology usually? So, and we actually spoke about how you do it. So those two questions are quite related. So if you do it with a CRO or you fully integrated it, probably would have an impact on how fast you can integrate it. But that's the point of this question. All right. Well, I guess we can go already to the discussion with the panelists. We have, I think there's 4 questions, well, 3 questions, but one that is just something I would like some of the panelists to bring forward a few experience of their own. So I know most of the panelists already use high-end technology to do their drug research. So I would like to, if you can, cite one of your experience where you actually have integrated new technology and how it has sped up, if you think about doing it the standard way, the discovery and going to the clinical side. So
I can comment a bit on some of the work that we're doing integrating biophysical methods with molecular simulations. And so historically, there's been kind of a one or the other. You run a calculation and then see how that compares to the experiment, or run an experiment and see if you can recapitulate that. Academically, that's super interesting. And that's somewhere where the early validation comes. But in order to really make an impact in drug discovery, it's really about that tight integration. And it's not just even a back and forth, but really, utilizing the computational data to drive the experiments, or using experimental data to drive simulations. So we found NMR to be particularly valuable, in the context of really understanding biomolecular recognition and mechanism. It really captures everything that we need from an atomic level to understand conformational mechanisms, MOA, et cetera, how your molecules bind, but it doesn't tell you everything about the structure. And that couples with what we're doing primarily in terms of molecular dynamic simulations and quantum mechanics simulations to really bound the problem. And we've been able to really make a lot of advances internally, and working with partners, CROs, and academic labs to really advance the methods to, again, that tighter integration where it's not just a back and forth, but really, the people from different labs and different groups sitting in the same room together, trying to figure things out, and that often requires new code development and many different players all sitting together, working on this. But we've got made a lot of progress on very challenging targets by taking this approach.
So, yeah, Woody, you have a company that's ultimately based on that principle, actually. Yeah. And what I hear is that, and I like to hear that, because innovative technology, you're speaking about people around the table. So this is it. I think it's something that is very important for such a technology to go from a concept that, like you said, was used in academia but was not pushed forward. The idea was to bring people around the table and say what is missing, to bring it into something that will actually build information to make a drug.
Yep, exactly. And sometimes that involves science. Sometimes it's just technology and automation and being able to take something that used to take, months and bring that down to a time scale of days or whatever is necessary to impact the project needs.
All right. That's a great example.
One of the things that we're doing is actually using multi-parameter flow cytometry. So, and we're doing that in a fully automated lab. So what we can do is take a specific patient sample and drug the sample with again, hundreds of different drugs and combinations. And using multi-parameter flow, we can look at 10 to 13 different cell surface markers to look at the effects of those drugs and combinations on specific cell populations within that sample to determine whether or not there are any changes induced in that are specific for the cancer cells versus the immune cells that might be present in that sample. And we're in the process of integrating a new flow cytometer that has 5 lasers, so we'll be able to look at 24 or 25 different cell surface markers to really define the entire sort of the immune population space really in great detail. And again, looking not only at sensitivity to single drugs, but then also test at high scale any number of different combinations that you might not normally think about. So we can actually take this do this in a very agnostic fashion that doesn't require any sort of mechanism, any sort of predefined notion around whether 2 mechanisms might be synergistic or not.
Another great example that includes automation, and I think at the end, bioinformatics gets pretty important once you are able to generate that much data. Adding a new laser adds a lot of possibility for in your case, and it's actually at the end a lot of more data that you will get. All right, so let's jump to the next questions. So what are the key factors an institution must have to successfully integrate new technologies? We want to speak about the pitfalls, the culture around it, and it's more philosophical questions than something that has a clear answer. But I would like to have your experience on that.
Well, maybe I can speak a little bit to get the conversation going. You said already culture, and I think that is the really key point. Innovation is never something that happens just as it stands. In particular, if a company is relatively large, there needs to be a culture that supports innovation. And that basically means that there need to be processes that allow innovation, that allow ideas to be brought up. Also, quite frankly, there needs to be a way of taking the fear of failure away from people who might be thinking of innovations. There need to be ways of assigning funds relatively fast, and some innovation can be done relatively cheap, but not always. There needs to be a feedback loop. So you don't want to have somebody running off and spending the rest of their paid time playing with their hobby in their garage. But you definitely want to give people space for innovation. For example, at Merck, we had an 80-20 rule where it was really, if you spend 20% of your time on innovation, that was kind of the standard. And 20% is a really good number in my opinion. You do need to have a mechanism to kill the things that don't work. It's the same thing with innovations as it is with drugs. Basically, if you let them run and they just don't work out, then you do basically lose opportunity for other innovation or just for regular work getting done, and you don't want to do that. So innovation does take passion, it takes persistence, and it does take good judgment. And this passion needs to be supported. And the good judgment belongs also in the category of communication. You want to be sure that you communicate with your innovators, that they focus on the problems that they see, and that they really are trying to keep traction and have an opportunity to really say, okay, this doesn't work. I want to do something else, or you know what, I wait for the next opportunity. And there must not be any punishment for saying that, so that we can really move on if it doesn't work. So that's just some thoughts about really supporting innovation in a culture. If that is not given, it's very hard for the innovators. And when they find that it is a fight, it's also easier to just not bring out the good ideas and to also not see when things don't work out. So just a few ideas on that.
Yeah, I think if people around here haven't read or listened to How Google Works, you're speaking about the 80-20%. That's a big mantra they had. But in that book they explain pretty well what is a culture of an innovation in a big company like Google. It's very interesting in that point.
I think there is one. Sorry, I think there's one point I would like to the excellent points made by Anka, and that is when we define innovation and all that, we in the pharma industry in particular are too siloed for our own good. I mean, if we take a new technology and then just assume it's good for discovery, and then the development people don't even hear about it, that doesn't bode very well. Uh, Much of the early advances in biophysics and all that, the process and other people just don't get to even hear about it, even though their company has had a stake. Similarly, if you go to all the sophisticated AI bits, where you are designing clever experiments, where you are scaling up, when you are designing retrosynthesis software, that is not made accessible to the medicinal chemist. So I'm a big one for calling for integration of technologies and other knowledge across the board.
Yeah, I think what you mentioned is, I don't know if it comes from there, but it seems that the model of having a platform, like you see in university, has followed into the pharmas. And the problem there is with platform, they tends to be over specialized in something and do not integrate the research community around. So that's a problem I see often. All right. Is there anyone else who would like to comment on that point?
I mean, I absolutely agree with Dr. Mukund and Anka. I think basically for any kind of new technology to innovate in large pharma, it kind of works both ways. Like, what is the service being provided on the other side, and how are we making or creating that champion? Because one of the key things that we have observed in pharma is like, it is very hard to find one single individual and ask them to be the champion, because it's hard. I mean, for them, it's less about even 80-20. It's more about their reputation, their time. So what we instead do is try to get in an early discussion with departments and help them understand what exactly are we trying to sell, or how we want to integrate, and then basically say, like, give us feedback. Tell us where you have concerns, where you don't understand what we are saying. And we try to, and it has been a good working model, especially for large pharma companies. To be very honest, with biotechs or even like big biotechs, this is not the problem. Their integration is more straightforward because the sales cycle is not long. But where there's real needed silos, where, you know, there's an HR department, financial department, VPs and directors, and the C-level folks don't really interact with each other, we try to make our discussions as easy to comprehend as possible, and then going deeper into the nitty-gritties of details that perhaps the scientists would really understand. So there's not one person who has to fully feel that, oh, I'm the only one responsible to get this information all around. And it is a lot of work from our end, because we have to put in more time. But I think if we want to innovate and create that kind of room in big pharma companies, I just don't see any other way to integrate unless we completely change the pharma model and ask them all to interact with each other and share the knowledge that's happening.
Yeah, I think on that point, there are pharmas that's starting to interact a little bit more.
Absolutely.
Yeah, you see it.
100%.
Recently, there's a big fund that has been launched for development of antibiotics and things like that. So
and even consortiums.
Yeah,
There's good initiatives on consortiums too. For sure.
Yeah, and to Anka's point of failure, I think a key point is that one has to be ready to fail and know when to cut your losses, and that's really hard because you're committed, especially when you have a champion, and at some point there's going to be a decision point, and people are going to have to come to an agreement of whether it's time now or whether it makes sense to do it later, and I think that's always a hard situation. And if it can be prefaced with the getting everybody ready for that failure, obviously hoping for the best and preparing for the worst, that tends to help smooth the process so it doesn't feel like it's anybody's responsibility to force something to work, because sometimes that's the whole point of innovation, is that nobody's done it before, and it's quite possible that it won't work, or maybe the timing is just not quite right.
It's a good point, because as scientists, we're not built to fail. We try to push and push and push until there's something that works. Mm-hmm. In a business, it's you have a different point of view. At some point you say, well, okay, now the budget is at its end. We need to take a decision and either inject more money or hand it there and build on what we've learned. And that's it. Yeah. Pretty good points we brought around the table. Next questions. This one is about AI and physical science. We know that it's more and more being integrated. We have good example and people around the table that are actually using it. But I've seen so many things. I've done things as well internally, and I know that some published paper are, let's say, overfitted. So the question is, well, can it be a solution by itself? And I would like people to discuss about the good, the bad, and the uglies of using this. So it's very important to bring that up.
Indeed.
Woody, do you want to go first?
Certainly can give it a shot. Yeah, the way that we typically think about AI and machine learning is that if there's a pattern that a human can detect and you want to do it faster, then AI is great. But if you don't know what you're looking for and there's no pattern, throwing a bunch of messy, sparse data at a program and praying for the best is kind of a risky endeavor. So I think there are some extremely good problems for machine learning and AI, and I think there's some really innovative work out there that's going on. There's companies that are generating the right kind of data in order to build models around. But in general, especially in the stuff that a lot of us are trying to do, which is novel targets and really trying to do things that others haven't done, by definition, that means there's probably not a lot of good data. And I think the vast majority of the efforts that we see in terms of publications are absolutely overfit, and you could show that over and over. So I think there's we're at the peak of the hype curve right now, and that doesn't mean that there's no value there. There's certainly some really, really good things out there, and we've got a lot of value out of machine learning. When you think of the right problem and you have the right data, it can be extremely valuable. But I think there's all these initiatives, and people are very compelled to just do something in this space because everybody's doing something, and it's very easy to get these beautiful looking models on training sets, but whether they're going to be useful in the real world where you're trying to do new things requires some thought. And it doesn't require typically a lot of thought in terms of deep machine learning algorithms and methodologies. The methodologies are pretty well established. Really understand scientific thinking and understanding your data and understanding what you're trying to make out of it. And if you're interested in interpolating between stuff that's in your data set and trying to recognize, reoccurrences of existing patterns, that's fantastic. And if you're trying to extrapolate outside of the existing data, or if your data is noisy, good luck.
I think, Woody, you make an absolutely brilliant point, as that is AI and all is an adjunct to helping you get better data, but ultimately is the human mind, the trained human mind, that is going to make the key decisions by proper understanding of the data. I have seen this far too often. People buy a robot or a robotic and then just disappear and hope all of a sudden they are going to get compounds like magic which are suddenly going to be active. There is still going to be needed a lot of active thought and human intelligence into doing some of those experiments and doing many of the things.
Yeah, so drug discovery is just too hard.
You made a wonderful point right now.
Yeah, drug discovery is just too hard for any one technology to solve.
Yeah,
it's a good analogy.
And the data that you put in is actually, incomplete. that you really don't have
Absolutely.
a clear understanding of all of the different pathways and interactions that may be occurring. And so, we actually see that in the clinical side, where, people are trying to extract clinical data and use machine learning to look at patterns. But, the data there are really dirty when you're talking about, they're based on billing codes as opposed to as opposed to actual diagnoses, right? That those billing codes are generated, for a very different endpoint than clinical accuracy. So you have to be very careful with the data sets that you're looking at.
It's
Right.
a very
And
good point.
I completely agree with Woody and all the other great points. And I think for anyone I don't believe that. First of all, AI is very general. There's a lot of different models within AI. Most people here already know that. And the choice of the model is also very critical outside of the data sets that's being used. Like, don't use transfer learning approach or autoencoders where they don't make sense. And it's the hard part about biology, because there's so many experimentations that are happening that may not even have correlation. So for example, if you're doing a cell line based assay and then you go in and do like even flow cytometry, really to find that correlation it's in itself a biological experiment where you need human intervention.
Absolutely.
So we have to creatively understand where, if and when we need to use machine learning, deep learning models, or even computer vision based modeling. But I think that is sometimes not core focus of many AI companies. And I think the more they will start working with biological data, they will start realizing it very soon that the data is sparse. There's not enough data. There's a lot of mislabeled data from human, animal. I think animals, we have more data just because it's less guarded in many ways. And even at the drug discovery. So finding ways to marry each other and then identifying where and if and when we need to use machine learning, deep learning models will really become the key. And I'm seeing that shift even in large pharma companies where they're realizing that, okay, we can't just inject AI and expect an answer unless you build a huge data set around that specific problem. Then yes, like how Woody mentioned, then it's definitely doable.
And I think for many people who got relatively late into the hype of AI or machine learning, which I want to remind everyone are two quite different steps actually. It's sometimes a little dangerous to just accept that there is a black box and expect those miracles to come out. When you have seen that grown up, so for example, in my imaging lab years back, we used to start with automated image analysis, automated imaging, and then automated image analysis. So that meant we suddenly were overwhelmed with the number of images, and the temptation was great to just really go into the automated image analysis. But it was kind of like getting a 2-year-old child in your lab that you really have to supervise all the time, because it turned out that you really have to look at the images. You have to correlate what your machine thinks it sees with what you know you're seeing. And you really also have to start understanding how these things come together. And when these steps are skipped simply because everybody tells me, yeah, AI is really not that transparent. Well, first of all, some algorithms actually still are relatively transparent, in particular if you run them on a relatively low level and a small data set. And it really helps to understand what you're doing, even if you are accepting a black box. I just want to remember that, because otherwise a lot of gobbledygook is produced.
May I take the liberty of asking our fellow panelist, and that is Dr. Hiroomi. You are representing something very interesting, and that is the clinical trials. What future do you see for AI in clinical trials? I mean, pardon this very layman's question, but can you use AI to predict whether or not one drug will be useful in this particular therapeutic area or not? I'm specifically asking this for the repurposing which is going on.
Mm-hmm.
So you bring that much needed expertise into this.
So it's interesting. So if you think about, there's a group called GCAR, the Global Coalition for Adaptive Research, that's running a multi-arm platform trial in in COVID, right, for repurposing as well as investigational drugs. And so they're, what they're doing is called response adaptive randomization, in which they'll learn from the experience and outcomes of every patient that's in the trial to inform future randomization of new patients into the trial. So even though it's not AI, it is based on a machine learning algorithm around how do you take those. How do you take the lessons learned from the patients who've already gone through the trial and use that information to inform how what are the clinical characteristics or what are the biomarkers that that characterize specific subpopulations of patients that may or may not respond to certain drugs. And we're actually going to use that in our own clinical trial, in which we want to take our platform using drug sensitivity and ultimately use that to learn what and identify what are the patient subgroups that are most likely to respond to certain sets of drugs or combinations. And again, it's using sort of response adaptive randomization to continue to learn from every single patient that comes through the trial, so that later patients who enroll actually benefit from that experience. And so I think that's one area in which novel clinical trial designs can really sort of push and enable drug development.
Very good. Thank you.
Mm-hmm.
All right. Okay, so let's move to the next part. I think we've done a great roundtable. Everyone has participated and brought very interesting point. So we are at the part where Sam, could you start revealing the poll answers?
Sure. First one.
All right. So yeah, well, apparently we have brought some bias into the answer, or that's the answer. So 86% of the response tells that they like to collaborate with CROs to integrate new technologies. A few of them says they will fully outsource it. And no one actually wants to jump in and just fully develop internally. I think that's the job of new innovative biotechs, I think.
Yeah, we're doing it, but I tried to press the button, but it wouldn't let us vote as panelists.
Yeah. All right. Okay. Next answers. What do you think will affect the most early drug discovery in the coming years? So, well, definitely computers, AI, anything related to data analysis, I think is very important. People totally lost hope on new instrumentations. That's okay. I think a lot of things are currently limited by physics laws. So new instrumentations, the gain are starting to be incremental. New methods, new ways of doing research, molecular biology and real world data are even there. And I think people still believe that technology like CRISPR, you never know, may appear again. And every 10 years you have something new that goes out from molecular biology. So that's very interesting. This one, all right. So where the innovative technology have the most impact? The speed of discovery, so overall speed of discovery. So not only a single thing, it's more a pipeline where if you integrate it, people think 50% of the people agreed on that. Biomarker still at 22%, and lead identification at 17%. Lead qualification, I think people still rely a lot on the standard ways of doing it. At the end, you need to put something to the FDA. So if it is a bit too fancy, I think the FDA will not agree with you. Mechanism of action, understanding, at around 6%, and people don't believe much in clinical trials right now. All right, last poll questions. 75% of the people agreed on months to integrate new technology, and if we remind us that the first question told that people actually integrated with CROs, and I think that's the only reason why you could say that, because otherwise, Woody, you correct me if I'm wrong, it takes years to integrate new technologies.
Yeah, sometimes it's a huge range. I mean, it really depends on the complexity. Some of our best wins have been very short 1-week projects where you get the right 2 people in a room that never realized that this person can use data to help this person and vice versa. And a half day of work has been transformative in some cases. And in other cases, you know, it can take years.
Yeah. All right. That was fun. All right. So we are at the end. We have still time for a small roundtable, just closing remark. Something that you would like the audience as a take-home message, that would be great.
Well, maybe I can just make a few remarks, and I think they are kind of in line with some of the things that I said before, simply because those are my mantra. And I have a favorite word, and that is balance. And I really think that innovation is definitely a balancing act. One thing that we didn't talk a lot about yet is that actually the risk in innovation is changing across the range what you do in pharma, from discovery all the way to development, where the later in the process you are with your innovation, the higher the risk. And therefore, I think it makes really good sense to start with innovation early on and bring it in the earliest you can. We also see a lot of that innovation being brought in by really larger pharma buying companies that took that first innovation step once they made it through it. So there are good reasons for that. And I also think that there is a temptation to take innovation for the new, really having that new thing that everybody talks about. And that's a very big risk. I think innovation is best when it is problem solving, when people really come across things that they see doesn't work, and really go about trying to make it work and innovate in order to solve problems. That is also one of the things where sometimes innovations can be quite small, and they don't always have to be very fancy big machines or anything. It can just be really a new way of doing things, or even a way in changing a process. And these should be appreciated because they quite often make a difference. So I just really want to advocate for bringing innovation in, having the guts to do it, balancing the risk by putting it in the right place and having the right communication and support around it. And then I hope it will really drive us.
But sometimes making something simple is an innovation.
Yeah, I'm happy to go next. I mean, I think for us, in order for our technology to be successful, we really need to demonstrate that there's clinical utility, right? That we're here developing drugs because there are patients who need new therapies. And so, for example, for Notable's platform, we need to be able to predict drug responses in patients, and we can also use it to drive the drug development process to help identify which patients might be most likely to benefit from those therapies. Ultimately, we want to be able to bring our technology platform to the clinic and hopefully enable physicians to identify the best therapies for their patients. Basically true precision medicine. And if we can't demonstrate, that the technology platform directly benefits those patients, then what we really need to be doing is something else that does. And so I think, again, as someone who's been in drug development, for the last 13 years, I mean, I think that's for me is the most important thing, is to make sure that there are direct patient benefits that come from the new technologies.
I can go next. I want to extend Dr. Hiroomi's thoughts to what we do at VSL. I think for us, we don't think there is drug discovery problem or drug development problem. there are several problems within this drug discovery and drug development, and everyone who is listening to the panel or even participating understands that. So finding one solution that fits for all is honestly really shooting in the dark and expecting great outcomes. So really trying to understand what the problem is we are solving is where the true combination of any sort of innovation will happen with science and or biology. And I think one of the key things, we are learning is the patience and understanding how this integration would look like, and being able to educate and make people aware while we thrive on several and levels of validation internally, externally, with their own data, with our own data, to really be able to not only show that, we are capable of doing it, but being more transparent where we are not capable of doing it, where we don't need computational resources, where a simple hands-on experiment would be important. So I think that level of transparency and openness would really, and it's definitely shaping the way our conversations are going on with the biotech folks and even at pharma. And I think this is going to be truly, we will see a new way of doing things within the drug development and even in the pharma industry. And I'm really excited about that. Thank you.
Yeah, I could say a few words. I guess the key balance, and Anka said this very nicely, is to be disruptive but not too disruptive. And finding that balance is tough. And sometimes things are going to have to break. But if you break too much stuff, it's going to be problematic for the organization as a whole. And people have to be aware that there's going to be a line where if you cross that line and it's being too disruptive, you may need to step back and wait some time, or you may need to pursue your innovations elsewhere. And it's sometimes very hard to change organizations, and innovation can be very scary to some people as it's uncertain, and uncertainty scares people. And with the right people and presenting things in the right way, typically you can get it done. But there's sometimes roadblocks that have to do with a lot more than just the science, and that often requires thinking outside of the box, and that may require thinking outside of the organization as well.
I guess I'm left over here still. What I would really like to see, I'm a huge fan of innovation. I'm a huge fan of encouraging innovation. I'm only calling for better integration of this innovation across the different disciplines, not in one fell swoop. May I give you a very simple example of this? I invented a chemical catalyst that mimics everything your human liver does, the cytochrome P450. One of my big differentiator was that since I work in organic solvents, I can detect and generate the metabolites which are unstable in water and which could lead to toxic side effects later on. We had an easier time to make metabolites on a large scale by and then do the characterization by mass spec. An unintended consequence was the pushback I received from small biotech firms. They said, "Hey doc, if we come to know that this metabolite of our one product could be toxic, it will seriously impact our ability to go and license this molecule. So let it be the other big pharma's problem," and so on. The last thing I would like to say is that I would encourage, and I belabor my students that they should not stick to the tried and trusted narrow path, and they should expand their horizons by listening to lectures and other panels and all that beyond their own expertise. And I can certainly vouch for that. In the 1 hour that we have talked, I have learned several new things myself. So you can teach an old dog some new tricks, and I exemplify that very nicely.
Well, thank you very much. I think what I will do to resume that, I took a few keywords, and that will be my take-home message. So one keyword I have here is a mission. So don't forget your mission. Keep it simple. Bring the experts around the table. Not mentioned during the panel, but mentioned a lot during the roundtable: quality. Quality of data, quality of everything, being transparent, balance, being bold, but not too much. Going outside, seeking outside expertise and integration. So those are the keywords that I had there, and those would be my words.
Yeah, well said, Hugo.
Thank you, Hugo. Great panel. Thanks to all the panelists for a wonderful discussion this afternoon to wrap up the summit. Just have a few closing remarks. It's been a good day, in my opinion. I hope you feel the same way. We began the day talking about stroke, and then we learned about HIF for cancer, and LCMSMS, glaucoma, ovarian cancer, and artificial intelligence. Looked at possibilities for new drugs for fibrosis. Discussed smart drug delivery and how it might apply to COVID-19. Looked at nano delivery across the blood-brain barrier. New ways to develop bispecific antibodies. Also looked at a hybrid model, also could be applicable with COVID-19. And then we just had this excellent panel with Hugo chairing on artificial intelligence. Let me say thank you to the Precision team, to Victor, to Sam, to Sherry, and thank you to all the audience that actively participated and made this a successful program. And with that, I pass it to you, Sam.
Once again, I want to thank first of all, our chairperson the entire day, and before this summit, he's been working day in, night with us, Sherry Monroe in marketing, Victor Sharif, head of operations, Luke Castro, Noah, our entire team, and all of our keynote speakers, our event sponsors, our panelists. Everyone worked tirelessly. Even though there are current restrictions in the world today, travel restrictions, restrictions on social gathering, we all still made the effort to share scientific thoughts and opinions, and for that we are very grateful. Hopefully everyone came out of this virtual summit with some sort of thought-provoking ideas, maybe even new business ideas as well. So feel free to follow our social media pages on LinkedIn if you're not, Twitter, follow our website for our future events as well. And we will be hopefully having more virtual summits more often. And we do have a final poll that I want for your feedback, most importantly, if you can all just check the screen. What areas can our virtual webinars improve on? You can select more than one answer as you wish. There's lots of options. This was our second virtual summit. We had our first clinical trial one back in June, which was a huge success as well. So please let us know your honest feedback. It's going to help us improve as an organization as we go forward, because we don't know when that COVID vaccine will be released. We don't know when we'll be able to organize an in-person meeting, which ensures the safety and health of all of our participants. So please click on the section. And yes, Alex, the sessions are recorded and they will be available. Do give us a week time span. We have an IT team that will be working on this recording, and we'll be posting it on our YouTube page as well for everyone to enjoy, hopefully in the future. So once again, thank you everybody for your feedback, and yeah, we'll all stay connected in the future. Reach out to me, Sam at precision-globe.com, Sherry or Victor at precision-globe.com if you need any other details. Thanks again. Stay safe, stay well. Speak soon. Bye.
Thank you, Sam. Cheers.
Cheers. Cheers.